Hello, everyone. Welcome to the WACBE webinars. Our Distinguished Biomedical Engineering webinar series is hosted by the World Association for Chinese Biomedical Engineers. My name is Julie Ji. I'm the Associate Professor of Biomedical Engineering at IUPUI, which is a joint campus between Purdue and Indiana University at Indianapolis. In terms of today's seminar, our speaker will present for about 40 minutes, and then we'll leave about 15 to 20 minutes at the end for a question and answer. Throughout the presentation, please remember to use the Q&A feature at the bottom of your screen in Zoom to submit your questions. So just type your questions there. We'll try to get to as many of them as possible after the talk during the Q&A session. So today we are very excited to welcome Professor Jiang. Dr. Jiang is a chair professor at the Southern University of Science and Technology in Shenzhen, China. He's been there since 2018. He obtained his BS degree at the University of Chicago in 1999 and a PhD at Harvard University in 2004. In 2005, he joined the National Center for Nanoscience and Technology. He was awarded the 100 Talents Plan of the Chinese Academy of Sciences, the National Science Foundation of China's Distinguished Young Scholar Award, and the Human Frontiers Science Program Young Investigator Award. He's a fellow of the American Institute of Medical and Biomedical Engineering, as well as the Royal Society of Chemistry. So we have a very distinguished speaker today. Dr. Jiang, welcome. If you are ready, I am going to...
Yes, I'm ready. Thank you, Julie, for that introduction.
I am going to turn this, stop share, and you can share your slides.
Thank you, Julie, for... Can you hear me, Julie?
Yes.
Okay, thank you very much for the very nice introduction. And I will take this opportunity to tell you about using microfluidics and nanoparticles for combating drug resistance. Drug resistance is a huge problem. I'm mainly going to talk about two sets of problems in medicine. One is the resistance against antibiotics. The second is resistance against chemotherapy in cancer. So let me start with the problem with antibiotic resistance. So human beings have really benefited quite a bit from the antibiotics. When human beings didn't have antibiotics, the average life expectancy was much lower, less than 40 years old. But when we started to have antibiotics, the life expectancy suddenly increased quite a bit because they kill the bugs. But recently, because of the abuse of antibiotics, drug-resistant strains of bacteria emerge quickly, and this is a global phenomenon. So if you look at the world map, the darkness of the green color indicates the level of drug resistance, and you can see that throughout most of the world, there is a fairly serious problem with drug resistance. So new types of antibacterial agents are really urgently needed. So about 10 years ago, we came across a set of gold nanoparticles that are very interesting, very unique. So gold nanoparticles by themselves are not antibiotic. So some of the audience may know that certain nanoparticles, such as silver and copper, they are antibiotics. The reason why they are is because elemental silver and elemental copper are very effective as antibiotics, and nanoparticles seem to enhance that effect. But gold itself is actually not antibiotic. And then we used a bunch of small molecules, small organic molecules, to modify the surface of gold nanoparticle. And this particular one, it's called diaminopyridinophile, which we abbreviate as DAPT. So by itself, this organic molecule cannot be used as an antibiotic. So it doesn't have any antibiotic property at all. But once you coat the gold nanoparticle with the DAPT on the surface of the gold nanoparticle, so this combination suddenly becomes a very effective and potent antibiotic. So we, and this, initially we found it to be very effective against gram-negative bacteria, such as E. coli and Pseudomonas aeruginosa. And not only is the particle very effective against the laboratory versions of this gram-negative bacteria, we also used multi-drug resistant strains of these bacteria. And we see that these particles are very potent against the multi-drug resistant versions of this bacteria as well. And we did something called the induction of resistance experiment, where you can culture the bacteria in concentration of the agent we're interested in at lower concentration than what is sufficient to inhibit the growth of bacteria. So typically lower half of the concentration of the half of the value of the minimum inhibitory concentration. So typically, if you do this in the presence of normal small molecule antibiotics, the resistance becomes very apparent within about 10 to 50 generations of culture. But what we found that was very unique in this particular case is that it doesn't breed any resistance even over after 200 generations of this induction of resistance, where you culture the bacteria at a low concentration of the nanoparticles. So we did a few mechanistic studies, and what we found is that the nanoparticles can enter the cell membrane and inhibit ATP synthesis activity. They could break the cell wall, and it could also suppress the subunit of ribosomes for tRNA binding. And but I think the most important is that nanoparticles can induce the permeability of the cell membrane and leakage of the nucleic acid. So there's really a number of mechanisms in which the nanoparticles work to kill the bacteria. So that's, I think, we think one of the reasons why it's particularly good for fighting against the multi-drug resistant, or the MDR version of the bacteria, because typically bacteria become resistant against drugs by overexpressing certain proteins, and it already has to compensate for the overexpression of proteins by sacrificing some of its normal functions. If an agent has multiple ways to combat the bacteria, it's very difficult for the bacteria to develop resistant mechanisms in all those fronts. So this is, we think, one of the reasons why it's so effective. So one of the questions you may ask is, well, is DAPT unique? Can you get other types of ligands to modify the surface such that the gold nanoparticle can become also antibiotic? And we did screening experiments, of course, and we combined the microfluidic screening that allows us to have a very high throughput, and we did find a number of ligands that allow the gold nanoparticle to become antibiotic. And what I mentioned here is that initially the DAPT modified gold nanoparticle is only effective for killing E. coli or Pseudomonas aeruginosa, which are both gram-negative bacteria. We're really interested in finding nanoparticles that also can kill gram-positive bacteria, and also potentially, if you could find good nanoparticles that could target different types of bacteria, that would be ideal. So the broad spectrum, so having one agent that's broad spectrum may not be necessarily the best choice, because that would tend to breed resistance. If you have several different agents, or even maybe one agent for one type of bacteria, that's I think really ideal, because if you know what kind of bacteria you're infected with and you treat the patient with that particular kind of agent, it's much less likely that you will develop broad spectrum resistance. So we actually recently did manage to find a system in which we can use a very similar set of ligands to coat the surface of good nanoparticle in order to adjust the gram selectivity. So we know that so initially the binding of the GAPT took place via the sulfur gold bound. And we know that amines could also bind to the surface of gold. But typically, when amines bind, it tends to form a lower density of ligands. So in one of the screens, we found this PBA, or phenylboronic acid, so molecules with this benzene ring with a boronic acid, fairly simple structure. On the other side has a thiol group. So these ligands can also activate the good nanoparticles to become effective as antibiotics. So we then try to look at the amino version of the same molecule. So basically, the phenylboronic acid portion of the molecule is exactly the same. So here is exactly the same. The only difference is that this molecule uses sulfur to bind to the surface of gold. This molecule uses amine. So amines tend to form much lower density ligands on the surface of gold compared to sulfur, which forms a high density ligand on the surface of gold. And we realized that we tested the particles against a very broad panel of bacteria, and we found that the sulfur-terminated phenylboronic acid could kill the gram-positive bacteria very selectively, whereas the amine-terminated phenylboronic acid is selective against the gram-negative bacteria. So I won't go into the details as to the discovery, but it's sort of an educated guess. Even before we started, we thought that there might be some differences in the ligand density having to do with the gram selectivity, but I won't go into all the details. But it turns out that the gram selectivity is actually quite good, and it's very, very safe. So that's the point that I think is very important. When you try to develop a drug or a therapeutic agent against a bacteria, you always have to consider the safety, and it has very high safety. And we also looked at whether or not you can form co-ligands on the surface. So basically mix the amine-terminated and the sulfur-terminated ligands on the surface. And then it turns out that we can actually adjust what kind of bacteria the nanoparticle can kill. So it's sort of a selective killing of the bacteria using gold nanoparticles. So if you look at the gram-negative bacteria, such as E. coli, MDR, the multi-drug resistant strain, Pseudomonas aeruginosa, and the multi-drug resistant version, and Klebsiella pneumoniae, and the multi-drug resistant version. So I already told you that the amine-terminated is very effective against the gram-negative, and then you can start by adding a little bit of the sulfur-terminated, and you can see that so these numbers are the MIC value, I should mention, the minimum inhibitory concentration. The smaller the number is, the more effective the the antibacterial agent is. So you want a small number. The smaller the number is, the more effective. When you go to these very big numbers, that means that they are no longer effective. So you can see that they transition into for the gram-negative bacteria, the sulfur terminated, or the chemical term is mercaptan, is typically larger than 64. So that simply means that if you, even when you're adding a very high amount of the reagent, the bacteria still keep growing. And for the gram-positive bacteria, so the opposite. So basically, the amine terminated are not effective, and the mercaptan, the sulfur terminated ones, are very effective. Whereas you can add in a few molecules of the amine terminated too, and you can it still doesn't affect the gram selectivity. So we did, again here, a few mechanistic studies. And it turns out that the amine-terminated gold nanoparticles targets lipopolysaccharides. So lipopolysaccharides are molecules that are present in very high concentrations on the surface of gram-negative bacteria. And then the sulfur-terminated nanoparticles target lipoteichoic acid, which is present on the surface of gram-positive bacteria. And we can look at how they interact in the transmission electron microscope, or the TEM, where you can see that the amine-terminated nanoparticles are aggregated by LPS, and then the sulfur-terminated nanoparticles are aggregated by LTA. At the moment, we don't completely understand how these different densities of the ligands on the surface actually bind to the surface, bind to these molecules selectively. One of the reasons could potentially be that these molecules have different spatial conformations, and we're still running molecular dynamics simulations to look at why these different types of conformation might result in different binding, even though they both terminate in phenylboronic acid, basically the same molecule, but different densities may allow them to have different three-dimensional configurations. So very much like when proteins form secondary and tertiary structures, even though some of the residues may be almost the same, the three-dimensional arrangement of these residues allow them to have very different binding affinities with other proteins and very different types of small molecules. So I mentioned that these particles are generally very safe. So we look at not only the MIC, but also the lethal dose, the 50% lethal dose of the LD50, and compare them with the commonly used antibiotics such as ampicillin, vancomycin, gentamicin. And you can see that the LD is actually very high. So in here, the higher the number, the better. The higher the number, it simply indicates that the particles can be tolerated at fairly high concentrations. So that's how the surface ligands actually may allow you to achieve different types of functionality using gold nanoparticles. So another parameter that can obviously be adjusted with gold nanoparticle is its size. We can synthesize gold nanoparticles of different sizes very easily. So here we again used the DAPT terminated gold nanoparticle, and we synthesized nanoparticles that have very different sizes. So the ones that I told you before are the 10 nanometer ones, so the ones that we initially discovered, which are only effective against the gram-negative bacteria, but not effective against the gram-positive bacteria. What we found out is that if you decrease the size of the gold nanoparticle, not only the gram-negative bacteria completely killed, but also the gram-positive bacteria also killed. But when you increase the size to about, say, 10 nanometers, you can see that the nanoparticles actually stop becoming useful as antibiotic completely. So again, here we tested against a panel of different types of bacteria. So when there's a low number of MIC, we highlight it as green to indicate its effectiveness. And when it's not effective, so we highlight that as red. So you can see again that the gold nanoparticle itself is not antibiotic. The GAPT itself is not effective. And then the ultra small, so the very small one, the 2 nanometer one is very effective, not only against the gram-negative ones, but also against the gram-positive ones. And that's not only useful for the laboratory strains, but also the MDR. And we not only looked at the MDR strains from ATCC, the standard, the microbiology banks, but we are lucky to get clinical isolates from local hospitals. And then these seem to be fairly effective. And for the gram-positive ones, you can see that the very small ones are quite effective. So we think for the very broad spectrum, the nanoparticles, one potential avenue for using it is for wound healing, because in that case, you don't really know what type of bacteria can become the infectious agent when the wound is healing. So I'll talk a little bit more about that later. And there's another way to actually change the core of the nanoparticle. So we not only looked at just one type of metal particle, but also alloys, so bimetallic nanoparticles. So we looked at basically gold doped with several other types of metals. Initially, we actually looked at gold-silver alloy to see if we could decrease the toxicity associated with silver. So I told you that silver nanoparticle is a fairly potent antibiotic. But the problem with silver is that silver itself, the metallic silver, is not only toxic against bacteria, but also toxic against mammalian cells. So to use it as a drug, its safety is a big concern. So initial thought is if we added the gold into the alloy to replace some of the silver in the nanoparticle, maybe that ought to decrease the toxicity. And we were successful to some extent, not as successful as we had hoped. But our students at the time were also playing with several other types of noble metals that could easily form alloys. For example, we looked at gold, platinum, AuPT, and gold palladium, AuPD, and gold rhodium. So basically, the gold platinum and the gold rhodium were the two that were fairly effective. And the mechanism we think here is a little bit different than the ones that I just told you about, where the surface ligands seem to play a very important role. In here, we think that because the nanoscale gold, platinum, and rhodium are known to be generators for reactive oxygen species, we think here by having several noble metals together may allow us to get nanoparticles that can generate the RLS to kill the bacteria. So there's another class of gold nanoparticles, which we actually call nanoclusters. So they're slightly smaller So they're even smaller than 2 nanometers. And these we call nanoclusters because they're not only smaller, but they have defined structures. So in principle, they behave more like molecules. They, in principle, have the same number of atoms of gold. And the other difference of gold nanoclusters are that they can be fluorescent. So they can fluoresce, whereas typically the gold nanoparticles are not fluorescent. So we also look at those, and we found a few that are also effective. So this is for particularly treating methicillin-resistant Staph aureus. So this is a hard to treat problem for many surgical procedures. Many orthopedic surgeries can have orthopedic surgeries, and orthopedic implants tend to have a high occurrence of MRSA. So you can see that these particles, they're fluorescent. They absorb at ultraviolet, and they are slightly red. So they can also kill bacteria fairly easily, and they also seem to not to breed any drug resistance. So this is an example where we looked at the MIC value of the gold nanoclusters, and you can see that it's very low over a course of 30 days, many generations. Whereas if you compare on the same condition with oxacillin, the MIC increases, indicating that the resistance has occurred. So we characterize the ultrastructure of some of these bacteria, and you can see that the bacteria tends to be deformed. If you look at these scanning electron microscopes, you can see that the bacteria tend to be deformed very easily in the presence of the nanoparticle. We can also look at the TEM, the transmission electron micrographs. You can see that the bacteria can be disintegrated. So the disintegration of the cell membrane structure, the cell wall structure in here seem to play very important roles. So we're able to also test quite a bit on animal models. So for example, the mouse skin infection model is where we cut the mouse skin open and infected the wound with bacteria and try to apply the either vancomycin or the gold nanocluster on the wound dressing and see how quickly the wound heals. And you can see that the healing is quite significant compared to the untreated. We also looked at the mouse bacteremia model, where we actually injected the bacteria into the bloodstream of the mouse. And if you don't treat the mouse, or if you just treat the mouse with an antibiotic that's not so potent, the mouse will all die within a matter of 32 hours. But if we treat with the gold nanoclusters, they all survive. And you can also look at major organ infections, major organs, and you see that they are really cleared of all the bacteria in the heart, the liver, the spleen, the lung, and the kidney. So that's a very good sign that the bacteremia has been completely reversed. So there are many ways in which you can actually make wound healing, the wound dressing for healing wounds. We can, for example, we use this electrospinning technique where we make polymer nanofibers into a mat and then get nanoparticles on them to treat the bacterial wounds. And the biocompatibility, again, is very high. You can apply a very high concentration of the nanoparticle. particularly the MDR bacterial infection can be very easily reversed. And in some cases, we can even lower the bacterial viability in a mature biofilm. So biofilm is a structure that bacteria form after they have grown quite a bit and excreted their own extracellular matrix-like material that has a mixture of enzymes and saccharides. And they tend to be very hard to treat because drugs cannot easily penetrate the biofilm. And in our case, because of the very high biocompatibility, we can use the nanoparticles in very high concentrations. So in some cases, we're quite successful in treating even mature biofilms. So I think that's all that I want to tell you about the GO nanomaterials-based antibacterial agents. So the key here is that they are non-toxic because they're actually made, so their components are non-toxic, not real toxic drugs. So for GO itself is very high in biosafety, and the ligands we choose, we can choose the ones that are, can be administered at very high concentrations. We think polyvalency effect may be in play here. Some of them may, of these particles, for example, the phenylboronic acid, they may bind to the surface of the bacteria in fairly low concentrations by themselves. Because the phenylboronic acids are known to bind to sugars on surfaces of cells, and including the ones on bacteria. But if they're present in many copies, that dramatically increases the binding affinity. And an advantage of the nanoparticle is that you can use many types of ligands to modify the surface to give it a very high level of functionality. So the next part, I will switch a little bit to our efforts in dealing with drug resistance in cancer. So there are many ways in which cancer cells and tumor tissues could develop drug resistance. One of the ways in which cancer cells develop drug resistance is to decrease drug influx. Another is to increase drug efflux. So particularly increasing drug efflux by cancer cells when they express these transporters that can selectively pump small molecule drugs out of the cells is one of the ways that cancer cells can escape therapy, and particularly chemotherapy, where we try to use a toxic agent to kill the cancer cells. So we have a fairly extensive program with microfluidics, in which we use microfluidic channels to make small nanoparticles as drug carrier. So here we typically, what we try to do is to use the microfluidic channel to adjust the flow, the behaviors of the flow, such that we have a lot of parameters to play with in making these nanoparticles. The particles that we typically make are done by nanoprecipitation. So using a solvent that dissolves the polymer fairly well, and then let it mix with a solvent that doesn't dissolve the polymer well, and at interface, because of difference in solubility, there will be precipitates. If you were to do this in bulk reactors, you would typically end up in a very broad distribution of sizes because you don't really have a very good control of the interface. So using microfluidic channels, because you can precisely adjust the interface, and there are many parameters you can work with, you can get very narrow distribution of nanoparticles, and that allows you to get higher efficiency in targeted therapy. So in this series of work, we mostly use PLGA. It's a biodegradable polymer, very common in in transplant, medical transplant, but it's also very common in drug delivery. and it's FDA approved for many applications. And we use doxorubicin, which is a drug that's used in cancer chemotherapy, and that's very likely to induce resistance. So initially, we dissolve the PLGA and dox in DMF, in organic solvent DMF, dimethylformamide. And we introduce that along with water. And because of the lower solubility of PLGA in water, we will tend to precipitate at interface. So because we use microfluidic channels, that allows us to really change many parameters of the flow. You can bend the channel, make the channel into a spiral, and of course you can design the channel using photolithography to have very complex shapes. That allows you to have many things to play with. But we are particularly interested in looking at if we can get a series of PLGA nanoparticles that have different hardness, and how the hardness of these particles may have to do with the drug delivery. I know many of you have background in mechanics, so I think you might be interested in this area. So the way we make nanoparticles that have exactly the same composition but different hardness is by the following strategy. So we use microfluidic channels, and then the PLGA is a relatively hard, so we call that a relatively hard material. So it's a relatively rigid polymer. And then there's this liposome that you can form, which tends to deform very easily. So when we have the PLGA within a layer of water and another layer of liposome, then we have a lot of water between these two layers. So that's a soft particle. And when we directly coat the surface of the PLGA with a liposome, there's no water in between. So we can do this particle fabrication very easily with a channel. I don't think I'll go into the detail as to why. But in here, this type of particle is what we consider hard. This is a soft because of the layer of water in between. And then in the PLGA, you can easily introduce drugs that allow you to look at how drugs can become effective as for therapy. And we use a very large number of techniques to characterize and really see that there is a layer of water between the core and the shell. So we use TEM, we use this fluorescent quenching. So there's I won't go into the details, but you just have to believe me that this has water. And we also use atomic force microscope to test, and indeed the ones that does have water is much softer than the ones that don't have water. So you can think about the ones that do have water as if it's a water-filled balloon that's easily deformed if you squish on it. But in here, it's just a balloon, the skin of the balloon directly covering a piece of rock. So it's hard. And we looked at how cells uptake them. So here you should observe the yellow fluorescence. So the more yellow fluorescence, the more drugs are uptaken. So the hard particles, this is a hard particle, this is a soft particle, and this is a particle of the liposome without even the inner core. And you can see that the hardest one actually gets uptaken the most. So we also did some animal experiments to confirm the cellular result. So then you may wonder, well, why is the harder particle more easily uptaken? So here we collaborated with Dr. Xinghua Shi, who was at the moment, he was in the Institute of Mechanics, but now he's at the National Center of Science and Technology. So what he told us is, so basically he ran a simulation because it's impossible to look at. These nanoparticles are less than 100 nanometers. It's very difficult to resolve them under the optical microscope, let alone look at its shape changes. So he ran the simulation. So I think after looking at the cartoon, you probably already realized that with a harder particle, when it enters the cell, it barely undergoes it doesn't undergo any shape changes. Whereas in the case of a softer particle, it has to undergo a shape change as it squeezes itself into the cell. So he tells us that the shape change is actually energetically very costly. And that's why it tends to enter the cell much less easily. So we really use this to test our capability to fight with multi-drug resistance. So in this particular case, we're looking at the drug efflux pump. As I told you, one of the reasons why they're made is because of the drug efflux pump can pump the drugs out to make drug-resistant cells. So we thought about, well, why don't we have a nanoparticle that has the small interfering RNA against the drug efflux pump at the same time the chemotherapeutic drug? Will that allow us to get a particle that could reverse drug resistance? So we use this chip that's a little bit complicated, but it allows us to do the work. So when you think about that, the particle that we actually had that has a water balloon, the three-layer structure that has water in it, that's perfect, right? The water layer could easily accommodate the small interfering RNA because PLGA is fairly hydrophobic. It doesn't encapsulate the small interfering RNA that well. But it turns out that that one doesn't have a very high uptaking efficiency, right? So we have to do something else. So now what we wanted to do is to get the aqueous layer within the PLJ. So that's actually very hard to do. But using the microchannels, it actually is fairly straightforward. So we make a reverse micelle first to encapsulate the hydrophilic with the charged ones, such as small interfering RNA. And then we coat it with a PLJ layer, and then we coat another layer of lipid on it. So the reason why this is very good to be to do on microchannels is because reverse micelles are typically very unstable. If you were to make them in bulk, they fall apart very readily. And if you were wanting to coat another layer of PLJ on it, it's almost impossible. But in here, because you're doing this very quickly, once you make the reverse micelle, you immediately coat it with PLJ layer and then another layer of lipid. And then this structure effectively freezes the structure to give rise to this double layer structure. Then you can encapsulate your hydrophobic chemotherapeutic docs in the PLJ and the small interfering RNA here. And it turns out that it's really very effective in reversing the drug resistance. In here, you can see that the nanoparticle that has both the small interfering RNA against the MDR, multi-drug resistant one, the transporter, and the docs is very effective in keeping the tumor size in check. So I think I'm running out of my time, but I just quickly go through another application using microfluidics to screen for nanoscale therapeutics. So we can use microfluidic channels to make these multicellular structures, these cell-cell interaction chips. And we use that to look at the interaction between nerve cells and cancer cells in pancreatic cancer. And we use that as a high content screening to screen the carriers against for small interfering RNA. And we can do this. We can do hundreds of thousands of this screening in microfluidic channels. So it's hundreds of times faster than directly doing this on animal models. And we were able to, again, get our co-workers in the hospital to provide us with the patient-derived xenograft model. And that shows you that the animal experiments basically shows you that the screening that combines the microfluidic channel and the small interfering RNA is actually very effective. And we also use this to screen for artificial carriers for CRISPR-Cas. CRISPR-Cas is very useful for biological research, potentially useful for clinics, but I think if you use a viral carrier, I think it has limited application in the clinic. So we're interested in using artificial carriers, and we're able to use microfluidics to quickly screen, and we're able to show this for a number of different systems. So at the end, I want to tell you that using microfluidics and combination nanoparticles, we actually use this for point-of-care testing, immunoassays, and nucleic acid amplification, and we were able to scale up with a company in China, and some of our products have already been approved by the Chinese version of FDA. So with that, I will close and say that the drug resistance with both bacteria and tumor can be a big problem. And we have written a bunch of reviews recently of using how you can use microfluidics and nanoparticles to combat these very serious problems. And I want to thank the funders and the people who did the work. And this is our group in Shenzhen. And Shenzhen is a beautiful city that is on the seaside and also has mountains to hike. So if you guys, after the coronavirus vaccines, we don't have to be quarantined for 14 days, you're welcome to visit us in Shenzhen. So with that, thank you again.
Thank you very much. Thank you, Professor Jiang, for this fantastic presentation. I enjoyed it very much. Before we get into the question and answer,
thank you.
Here we go. I would like to call your attention to our next WACBE webinar on Saturday, November 14th. We're going to have Dr. Bing He, Trustee Professor and Head of the Department of Biomedical Engineering at Carnegie Mellon University, as he talks about EEG-based dynamic brain imaging and brain-computer interface. So make sure you mark your calendar for this next talk. As you can see on this slide here, we have a series of excellent speakers coming up. So I hope you'll continue to come back and join us for these webinars. Please also follow Columbia BME on social media to receive the latest updates. You can see their social media information here. You can also learn more about WACBE at wacbe.net. So make a note of these social media points. With that, I am going to switch over to the question and answers. So I'm going to go with the order that I see them. From Guiren Wang, we have a couple questions. What is the mechanism that the particles can emit fluorescence? I think in one of your slides you talked about some of them being fluorescent. And also, is the model dispersed particle size, nanoparticle size, important or not in terms of drug effects?
So can you still hear me?
Yes, I can hear you.
Okay. So the fluorescence, so only the very small gold nano clusters have fluorescence, and the ones that, and they should have also finite structures. And the fluorescence has to do with how the small structures, that very confined structure, changes the energy landscape. And the way the electrons bounce around will allow them to emit fluorescence. So that's a short answer. So the size of the particle actually have a very significant effect on drug effects. The first part, where the gold nanoparticles were used, the very large particles, the particles that are larger than 20 nanometers are not effective at all as antibiotics. And the medium size, about 10 nanometer ones, are the DAPT modified, one of the first ones. It's only effective against the gram-negative bacteria. Whereas when you decrease the size to about 2 to 3 nanometers, they actually can be effective again to both gram-positive and gram-negative bacteria. In the second part, where we use the PLJ nanoparticles, the size actually has a profound effect. It has to do with how long the particles can be circulated in the blood, and how quickly they can be cleared. The very small particles tend to be cleared by the kidney, and the slightly larger ones tend to accumulate first in the liver. So also, but that also has taken into consideration the surface chemistry and the ligand. So it's not a very straightforward answer, but for error. So the simple answer is yes, but depending on the situation, it has a different effect.
Thank you. From Si Hong Wang, a couple questions. Your gold nanoparticles suppress binding of ribosome with tRNA. Is it specific to bacteria or also in mammalian cells too?
Yeah, that's a very good question.
Yeah.
We haven't actually tried on the mammalian cells because the particles don't actually enter bacterial cell, but we should probably try that. It's a very interesting question. At the moment, we only know that it binds to the bacterial ribosome.
Okay. And also, how did you deliver your PLGA nanoparticles in the animal models?
We typically use tail vein injection.
Okay. Question from Dr. Wu. Whether the degradation rate of PLGA matters in your application. If so, has your team looked at it, the degradation rate of PLGA?
Yes, it does. We haven't done it systematically because the degradation rate has to do with the drug release as well as the stability of the nanoparticle in the bloodstream and the clearance, I think. We haven't actually done that in a very systematic manner. So that's a very good question. Maybe we should do that more systematically.
Thank you. And from BMA, does surface charge affect or improve the gold nanoparticle efficiency?
Yes, that's also something that we look at very carefully. So if the surface charge is too, so generally the very positively charged surfaces have better antibacterial properties. But the problem is that when the surface charge becomes too high, it also becomes toxic against mammalian cells. So we have to play a little bit. So ideally, we want something that's not toxic against bacteria, not toxic against mammalian cell at all, but can kill bacteria. So what we found is that the slightly positive charged ones and the few even negative charged ones can kill the bacteria, but without affecting the mammalian cells. But surface charge is definitely critical.
Thank you. And from Yalin Liu, I think, would protein in body fluid absorption on the surface coating influence its antibacterial or cancer targeting performance? Also, would soft nanoparticles have different transport properties in blood?
Yeah, those are good questions. So the gold nanoclusters, the very small gold nanoparticles, they don't seem to attract so much proteins on them. So when they're injected, so that's our preliminary finding. And it seems that the bigger nanoparticles the gold nanoparticles tend to recruit proteins after they enter the bloodstream, but we haven't completely been able to understand what types of proteins have absorbed because our ability in proteomics have not been able to give us a repeatable answer as to what kinds of proteins are on there. For the softer particles, yes, the bloodstream actually the bloodstream actually plays a very important role because do keep in mind that there are lots of cells, white blood cells in the blood, and they also tend to uptake nanoparticles. And sometimes if the particles get uptaken too easily, they are cleared completely in the blood before they reach the target organ. So sometimes it's important. So the in vivo experiment it's not exactly the same as the in vitro cell experiment. So you have to be careful and choose your hardness carefully.
Thank you. Questions from Sarah O'Connor. Very interesting that the smaller the nanoparticles are, the more antibiotic capacity they have. Does the size also affect mammalian immune responses? This will be relevant to the nanoparticles encapsulating the CRISPR-Cas9. If you can comment on that, that'd be great.
Yeah, good question. We haven't done that. We haven't looked at how we're doing that right now, but we haven't gotten anything meaningful to report yet. But that's definitely something that we're looking at. And you're certainly right that for the carrier for CRISPR, the immune system has to be very carefully looked at. Thank you.
I think we have one final question. The half-life of your nanoparticles in liver and kidney?
The gold nanoparticles, we have looked at some of them, and they tend to be less than they tend to fall within 1 to 3 days.
Okay.
The PLGA, we haven't looked at them in detail, but the PLGA particles tend to be in the liver for a longer period of time, maybe 3 to 5 days.
Thank you. One last question from Zhonghui Hong. How does the gold nanoparticle bind to ribosome, bind to protein or RNA? Ribosomal RNA.
Yeah, we simply don't know. We have not been able to, get a crystal structure or, yeah, we don't know. That's a hard question because the ribosome is such a complex machinery, and we don't know how it binds. So we don't have well, if anybody has the capability to look at, we'll be very happy to collaborate. That's a very important question to answer.
Thank you. We have a few minutes. I have a final question. You mentioned earlier something about implantable delivery systems or implantable devices. How practical is this method or this technology in terms of coating materials that can be implanted?
Yeah, so I think the biggest problem is still safety. Even though we show that the gold nanoparticle is quite safe, I think to eventually use it in the clinics, you have to do a lot of experiments to answer questions of half-life and residual amount and how that residual amount of material might change your immune system. So I think it's possible if we have someone investing a lot of money to look at these problems, these clinical problems, because the implantable. My understanding is that implantable devices still has a fairly high rate of infection, even a very long time after implantation. I've spoken to quite a few orthopedic surgeons, and they all agree it's a problem. Even though occurrence rate is not high, but it's still a big problem. If it occurs, sometimes you have to do the implant again. Sometimes it's even more serious. You have to do amputation and stuff. So we hope that it can eventually be used, but and we are working with some of those people to see if we can use this coating to better the treatment. But the problem is that because the occurrence is not high, and it's not easy to do the experiment because we can easily do the exogenously introduced, but some of those people tell us, well, it's different than what really happens in the patient. So we're still thinking about better models. If you have any good better ideas, let us know. We're happy to collaborate again.
Excellent. So I think we have answered all the questions. I think we're right on time. So I would like to thank Professor Jiang once again for an excellent talk and for your time to answer all the questions. And thank the audience for attending the webinar today. Make sure you guys register for our next talk on November 14th and enjoy the rest of your weekend. Thank you very much.
Thank you, Julie, for hosting my talk.
Thank you, Xingyu.
Thank you. Yeah, thank you. Fantastic talk. Thanks.
Yes.
Good night.
Good night to you.
Thank you. Good night.
Bye-bye. Bye-bye.
Bye-bye.
Have a good weekend.
Have a good weekend.
Bye.
Thank you.
Bye.
Bye.